The phrase "use it or lose it" is probably the most important study tip that any student can be given. And the purpose of my mind map here is to show the biochemical or neurochemical basis of use it or lose it with long-term potentiation. So the story begins here in the hippocampus, where signals from other areas of the brain enter the CA3 region, which then passes them on to the CA1 region. So we'll zoom in here. Have this CA1 neuron receiving via its dendrites signals from all these different CA3 axons. And then we'll zoom into this synapse here. We see that the medium through which action potentials that go down the CA3 axon are translated to signals in the CA1 neuron is via glutamate. It's an excitatory neurotransmitter that can bind to these two different types of receptors. They're ionotropic receptors, NAMDA and AMPA. And you see down here that glutamate binds AMPA. It allows sodium ions in because it's an ionotropic receptor. The effect of these sodium, or positively charged sodium ions, is depolarization. There's a resting negative potential across the membrane. Here the inside is relatively negative compared to the outside. And when sodium enters, that depolarizes it, and if the depolarization reaches a certain threshold, that sends an action potential down the CA1 axon to be passed on. Now, the part that's relevant to long-term potentiation is here with the NAMDA receptor. Glutamate can bind it, but it's currently blocked by this magnesium ion, so the channel is blocked. So what happens is, if enough signals are being transmitted, if enough glutamate is being released from the presynaptic neuron and binds enough AMPA receptors for long enough, enough sodium is admitted to electrostatically repel the magnesium ion, which then opens up the NAMDA channel. It can also let in sodium, but most relevant here, it can let in calcium ions. And this has many ramifications. Once calcium is in the cytosol of the CA1 neuron, it can bind to calmodulin, which undergoes a conformational change. It exposes the binding regions for both protein kinase C and calmodulin-dependent kinase 2. When it activates, or it binds and activates these two things. When PKC is activated, it mobilizes these passively stored AMPA receptors to the membrane, making the postsynaptic neuron more responsive to glutamate because there's more of these receptors for it. CAMK2 autophosphorylates when it binds calmodulin, allowing it to stay active even after calcium levels have dropped and calmodulin is deactivated. CAMK2 binds and phosphorylates AMPA, which greatly increases its ability to transfer sodium ions, which further strengthens the synapse by allowing an even stronger depolarization for the same glutamate binding. Then, and this here was early LTP. Now we're moving on to late LTP. If there's both a buildup of this activated calmodulin from there being lots of calcium admitted, and the binding of dopamine to the D1 receptor, these together activate adenylyl cyclase, which converts ATP to cyclic AMP, which acts as a messenger that activates protein kinase A, which goes to the nucleus and activates a transcription factor called cyclic AMP response element binding protein, CREB, which binds to the cyclic AMP response element on the DNA and causes transcription and translation of both more AMPA and NAMDA receptors, which strengthen the signal, and the expression of brain-derived neurotrophic factor, which is exocytosed and can act as an autocrine signal or a paracrine for other nearby neurons, binding to TRKB, which through another pathway that would be worthy of its own presentation causes the creation of more dendritic spines, which are spots for other CA3 axons to form synapses with. So the signal has been even further strengthened. The early LTP corresponds most strongly to the use it or lose it principle. If you have frequent signals coming here, either a lot from one neuron or the summation of many different signals from different neurons, you're going to get that level of depolarization required to kick out the magnesium ion more often, meaning you'll have these cascading effects of having more AMPA receptors and more strongly activated AMPA receptors responding more strongly to stimuli, thus strengthening the signal transmission to the CA1 neuron. The late LTP, also because calcium's involved, calmodulin, it also corresponds to the use it or lose it principle, but it also helps explain this little bit of advice that an old coworker gave me, which was that if you want to learn something, you either do it over a long period with spaced repetition or over a short period with dramatic impact. And this is sort of the role of dopamine here, is sort of the dramatic impact. If a memory is being formed while dopamine is surging, it's going to be a memory of something very emotionally impactful, emotionally salient. And through this mechanism that actually causes permanent changes to the dendrite itself, making it physically more receptive, you get strengthenings of the pathways involved in that memory. So yeah, that's long-term potentiation. That's the physical basis of use it or lose it and dramatic impact for memory formation.